Thank you, Gabriel, for the introduction. And I just wanted to take a moment to thank Synthase for allowing me to present some of my work today in the form of this webinar, and thank you all for attending. So without further ado, I think we should get started here. So just a little bit about me, as Gabriel sort of mentioned, I've had a long journey at Brock so far. I did my biomedical sciences degree as an undergrad at Brock University, and then moved into a master's of applied health sciences. After that, I did work for my supervisor, Dr. Val Fajardo, as a research assistant, and sort of figured out if research is where I wanted to be. And sure enough, I did stay at Brock under Dr. Fajardo's supervision, and I'm now starting my first year of my PhD in applied health sciences, still at Brock University. So this is our lab at Brock. We are located in the Cairns Family Health and Biosciences Complex. And as mentioned, I'm under the supervision of Dr. Val Fajardo. And our lab is the Muscle Plasticity and Health and Disease Lab. So in our lab, what we look at is sort of muscle in different disease states, such as muscular dystrophy, and how muscle can actually adapt to things like atrophy or changes in the environment, such as spaceflight. So another part of muscle that we actually look at is muscle physiology and how it actually relates to muscle metabolism or energy use. And this is what my area of research sort of focuses on, is how muscle is involved in energy expenditure. So today we're going to talk a little bit about our recent publication in the Journal of Biological Chemistry. So what we looked at was low dose lithium supplementation and how it can promote sort of energy expenditure through the SERCA pumps, or the Sarco endoplasmic reticulum calcium ATPase in muscle. So this was actually my master's thesis. And in our paper, we sort of coupled this to the work of another student in Dr. McPherson's lab, who's one of our collaborators at Brock, and her name is Chantel Ryan. I just wanted to give her a shout out as well, because she looked really at the adipose side of things. So we sort of took our two projects and put them together into this one publication. I did actually include a QR code here as well, so feel free to scan that, and it sends you the link right to the paper. So I guess we can kind of get into it right now. Beginning sort of with a big picture issue, which is obesity. So obesity and overweightness have sort of become a growing health concern across the globe. And as of 2015, approximately 1/3 of the global population was actually reported as overweight or obese. And obesity is this sort of multifactorial disease where individuals with obesity are at increased risk of developing different comorbidities, such as type 2 diabetes, Alzheimer's disease, cardiovascular disease, hypertension, and some other related disorders. And strategies that are sort of currently in place to combat obesity aim to mitigate the energy imbalance that's seen behind its pathology. So obesity is often a result of a chronic imbalance between caloric intake and energy expenditure, where caloric intake supersedes our total energy expended. And this sort of results in this increased adiposity state or increased fat mass. And cellular pathways that can actually increase energy expenditure have become of particular interest to us in the effort to combat obesity, in order to sort of maintain a balance between that caloric intake and our energy expended. And exploiting these cellular pathways that sort of contribute to energy expenditure could allow for a shift in the balance between caloric intake and energy expenditure, where we could further increase energy expenditure to outweigh caloric intake, resulting in reduced adiposity or reductions in our fat mass. And one of the pathways that is actually able to increase energy expenditure is adaptive thermogenesis. So total energy expenditure can best be described as the amount of energy that we use in a day. So taken together with energy intake or food intake, this can sort of determine our overall metabolism or metabolic rate. And total energy expenditure can be broken down further into energy expended by performing work, physical activity, and obligatory energy expenditure, along with adaptive thermogenesis. In adaptive thermogenesis, or the regulated production of heat, is this cellular process that, in response to cold exposure or caloric excess, energy expenditure and heat production is increased. And this results in the greater combustion of metabolic substrates and fuels, such as fats or carbohydrates. And it's actually been estimated that adaptive thermogenesis represents approximately 10 to 15% of our total daily energy expenditure. Therefore, targeting this process is like a good potential to increase overall energy use in the context of obesity and sort of mitigating that energy intake versus energy out imbalance. So within the body, there are two sites that adaptive thermogenesis occurs. So the first site of adaptive thermogenesis is within the adipose tissue, specifically in the brown and beige adipose through this protein known as uncoupling protein 1. And there are three types of adipose tissue. So the first is our white adipose tissue, and white adipose tissue has very large lipid droplets, little mitochondria, and this is known as our sort of typical storage fat. Brown adipose, on the other hand, has numerous smaller lipid droplets, which is not really ideal for storing fat, has plenty of mitochondria and a protein known as UCP1. And this protein actually facilitates thermogenesis, making brown fat this very highly metabolically active tissue. We also do see this sort of intermediate phenotype, which is the beige adipocyte. And this is a white adipocyte that's actually adopted a brown-like phenotype through inductions from the environment, like metabolic stressors or cold exposure, for example. And the beiging of white adipose tissue has become of interest in the context of increasing energy expenditure due to the surplus or the amount of white storage fat that we as humans possess, and the potential of converting it to a more thermogenic site like the brown adipose. So when looking at UCP1 in brown adipose, first we kind of want to see what the mitochondria and what the adipose tissue does in brown adipose. So the mitochondria that are found in the brown adipose are responsible for generating energy in the form of ATP through the electron transport chain and oxidative phosphorylation. So oxidative phosphorylation uses different metabolic fuels and results in the release of protons to the intermembrane space, which are then pumped back through the ATP synthase to actually generate ATP or energy. Although in the presence of UCP1, what happens is these protons that are within the intermembrane space can actually sort of leak back or enter back into the matrix through UCP1, and the energy that's released from doing so is actually released or expelled as heat. So if we do have more UCP1 or a greater expression of UCP1, more fuel or more energy will be required to input into that electron transport chain and oxidative phosphorylation in order to maintain the mitochondrial gradient as well as ATP synthesis. So now moving on to our second site of adaptive thermogenesis. So our second site is within the skeletal muscle. And the skeletal muscle can increase energy expenditure first, what's known through as shivering thermogenesis. So shivering thermogenesis occurs through sort of repeated contractions of the muscle. And with every subsequent contraction, there is a little bit of heat released in response. And we can't shiver forever. So once shivering thermogenesis ceases, we do have this mechanism called non-shivering thermogenesis beginning. And skeletal muscle non-shivering thermogenesis is actually mediated through the Sarco endoplasmic calcium, Sarco endoplasmic reticulum calcium ATPase, or SERCA for short. So in the skeletal muscle, SERCA actually plays a critical role in the excitation contraction coupling cycle by initiating muscle relaxation. And SERCA facilitates muscle relaxation through this active reuptake of calcium into the SR lumen. And in doing so, it sort of maintains these intracellular calcium levels and ensures that there's enough calcium available for subsequent contractions. So following the muscle contraction, cytosolic calcium levels are high, and this allows for SERCA to bind to calcium ions within its binding site. And calcium binding and the subsequent hydrolysis of ATP actually allows for this energy release and release of calcium back into the SR storage site. So since SERCA actually requires ATP utilization or energy use for mediating muscle relaxation and maintaining these intracellular calcium levels, it's thought that SERCA actually contributes a significant amount of energy to our resting metabolic rate, as well as our overall metabolism. And this sort of energy rate has actually been estimated to be around 40 to 50% of our resting metabolic rate and around 25% of our daily energy uses. So under optimal conditions, SERCA transports 2 calciums into the SR for every 1 ATP hydrolyzed, which we saw on our previous animation. And this is known as SERCA's optimal coupling ratio. Although in vivo, SERCA transport is much less efficient, thereby lowering our coupling ratio. So think of it like a fuel inefficient car that requires more gas per kilometers traveled. An inefficient SERCA pump with a lower coupling ratio will require more energy to regulate the amount of calcium levels in our muscles. And one of the factors that actually contributes to this is the presence of SERCA uncouplers. And these uncouplers, known as sarcolipin and neuronatin, can actually interact with the SERCA pump, making SERCA less efficient at pumping calcium. So sarcolipin and neuronatin, these uncoupling proteins, are able to interact with the SERCA pumps and alter accessibility to those calcium binding sites. So when calcium tries to bind in the presence of sarcolipin or neuronatin, it will sort of slip out, although ATP hydrolysis has already occurred. This in turn will lower SERCA's coupling ratio, meaning essentially that there's less calcium transport into the SR for every ATP hydrolyzed in the presence of these uncoupling proteins. So first, taking a look at sarcolipin. Sarcolipin is a small uncoupling protein that we know lowers SERCA efficiency, thereby increasing the energetic costs of calcium pumping. And it's actually been shown that mice that are lacking this sarcolipin protein are actually more susceptible to diet-induced obesity and show greater weight gain than their wild-type counterparts. On the other hand, mice that overexpress sarcolipin or have an excess amount of this protein can actually attenuate weight gain when placed on a high-fat diet compared to wild-type. Further, if we take into account total or daily energy expenditure, we can see that sarcolipin null mice, or mice lacking this protein, actually have lower daily energy expenditure compared to wild types. And this suggests altogether that sarcolipin might play a critical role in energy expenditure as well as weight gain, and potentially this is through SERCA uncoupling. So like sarcolipin, neuronatin has actually been recently identified as a SERCA uncoupling protein in muscle by one of my fellow lab mates, Jessica Braun, pictured here. And what we see is reductions in calcium uptake as well as SERCA's apparent coupling ratio when we have increased content of neuronatin relative to SERCA. So you can see with higher neuronatin present, we have a greater impairment in calcium uptake and greater alterations in coupling ratio. So this suggests that in the presence of neuronatin, SERCA calcium transport efficiency is significantly reduced. Neuronatin is also expressed in a wider variety of muscle types, including soleus, EDL, red gastroc, white gastroc, as well as other tissues in comparison to sarcolipin, which is found primarily in slow oxidative fibers, as well as muscles like the soleus and diaphragm. So this sort of makes neuronatin potentially an even more attractive target to actually increase whole body energy expenditure. So further, looking at neuronatin, it's been previously shown that neuronatin knockout mice actually have lower daily energy expenditure compared to wild-type counterparts. And these neuronatin knockout mice also have increased weight gain when placed on a high-fat diet. We have also recently obtained these neuronatin knockout mice in our lab. So this is the same neuronatin knockout colony, and we just did some DEXA scans on these mice last week. And based off the first round of scans, I know I don't have anything to compare it to, but it looks like these neuronatin knockout mice actually have greater body weights and potentially fat mass than their wild type counterparts. Although this is speculative for now, but it does just visually and sort of looking at the body weights appear that there might be differences there. So with that being said, we sort of sought to examine potential upstream regulators of SERCA uncoupling in the muscle, in the adipose as well, in order to increase the expression of these proteins, including UCP1, and therefore increasing our overall energy expenditure. So one of these potential regulators is GSK3. So GSK3 is a constitutively active serine threonine kinase, and it exists in two isoforms, GSK3 alpha and GSK3 beta. And it's most well known for its role in regulating glycogen metabolism. And GSK3 has actually been implicated in a number of different disease states and a number of tissues in the body, including heart failure, bipolar disorder, cancer, diabetes, certain inflammation states, Alzheimer's disease, and aging. And it's actually been, recently identified, like maybe this does actually have a role in obesity as well. So GSK3 may have this negative role in obesity, therefore its inactivation has become of particular interest to us, and it's possibly a molecular target in increasing energy expenditure relative to the obese state. So the inactivation of GSK3 actually occurs through the phosphorylation of certain serine residues, so serine 21 in GSK3 alpha and serine 9 in GSK3 beta. And this actually prevents substrate recognition by blocking the binding pocket and blocking substrate recognition. So GSK3 does need to bind sort of primed or phosphorylated substrates. Therefore, when GSK3 itself is phosphorylated on these serine sites, it actually recognizes itself sort of as a pseudo substrate, thereby blocking any activity. So since GSK3 may have this negative role in obesity, inactivating GSK3 as a potential upstream regulator of uncoupling and adipose thermogenesis presents a unique target to increase energy expenditure. So it actually has been shown that GSK3 negatively regulates the thermogenic program in brown adipocytes. So its activity actually inhibits the expression of genes that are involved in adipose thermogenesis, including UCP1, which is that uncoupling protein. On the other hand, reduction in GSK3 activity actually activates thermogenesis. We see that through increased oxygen consumption, which is a measure of energy use in brown adipocytes, suggesting then that the inactivation of GSK3 may actually be favorable in promoting thermogenesis in brown adipose cells. It has also been demonstrated that the overexpression of GSK3 in male mice actually results in increased body mass and fat mass, further providing the role or the potential role of GSK3 in obesity and in that obese phenotype. And at the time, while we could see the potential of GSK3's involvement in adipose-based thermogenesis, to our knowledge, it wasn't yet determined if GSK3 was also a negative regulator of muscle thermogenesis through SERCA uncoupling. So how is it possible that GSK3 influences SERCA uncoupling? Well, GSK3 is known to oppose calcineurin signaling. So calcineurin is a phosphatase, while GSK3 is a kinase enzyme. So while calcineurin promotes the dephosphorylation of this transcription factor known as NFAT, permitting its nuclear entry into the cell, GSK3 actually promotes the cytosolic accumulation of NFAT through its phosphorylation. And calcineurin and NFAT signaling has actually been implicated in the expression of different oxidative genes, including the uncoupler sarcolipin. So therefore, active GSK3 has the potential to reduce muscle-based non-shivering thermogenesis by actually lowering sarcolipin expression. So we sort of proposed that active GSK3 does have that potential to reduce muscle-based non-shivering thermogenesis by lowering sarcolipin expression and potentially SERCA uncoupling. because if you have less of an uncoupler, you'll probably have less uncoupling. Therefore, what we sort of hypothesize is that the inhibition of GSK3 would actually result in an increase in sarcolipin expression, and the subsequent increase in SERCA uncoupling, and therefore increased energy expenditure. So to explore the effects of GSK3 inhibition on both SERCA uncoupling and adipose-based thermogenesis, what we sought to inhibit GSK3 with was lithium. And lithium is a known GSK3 inhibitor. It's commonly used for the treatment of bipolar disorder. And lithium can inactivate or inhibit GSK3 for the phosphorylation of GSK3 on those inhibitory serine sites, thereby inactivating it. And it can also compete with magnesium, which is a cofactor for its activation. And our lab has actually recently demonstrated that low doses of lithium are able to inhibit GSK3. So what is the significance of this low dose treatment? So to avoid toxic effects that are associated with lithium treatment at the higher doses, it must be used within a narrow therapeutic range of 0.5 to 1 millimolar serum concentration. And we've actually shown in our lab that a low dose of 10 milligrams per kilogram per day does actually result in a serum concentration of 0.02 millimolar, which is well below that recommended therapeutic dose. So to this extent, what we essentially wanted to see was this relationship between low dose lithium. Does it actually inhibit GSK3? And does that inhibition allow for adipose or muscle-based thermogenesis?
Thank you, Mia, for the great discussion so far. Before we continue, we would like to ask a couple of quick poll questions, and the questions are the following. So the first question is, have you used DEXA imaging in the past? And the second one is, what is the main focus of your research? We'll wait a few moments to let everyone answer the questions, and please select the most appropriate response. We'll just wait a few more seconds. Just a couple more seconds and we'll move on. Thank you for answering the poll questions. I will now hand over the presentation back to Mia.
Thank you, Gabriel. All right, so we'll continue. So as mentioned, to this extent, what we did was examine the effect of low dose lithium treatment on GSK3 inhibition in both adipose-based and muscle thermogenesis. First, using a cellular model sort of as a proof of concept in both muscle and adipose. So here we treated muscle cells, C2C12 myotubes, and 3T3L1 adipocytes with a dose of 0.5 millimolar lithium. And following this sort of proof of concept in cells, we actually utilized some in vivo measures in a male C57 mouse model, as well as some ex vivo analysis of the muscle and adipose tissue from these mice. So for both muscle and adipocytes, cells, as mentioned, were cultured with a control and a lithium treated group, and those received the 0.5 millimolar dose of lithium chloride. So to first touch on some of our experimental methods in muscle cells, we performed methods of respiration to quantify cellular energy expenditure. So essentially this was looking at oxygen consumption in the cells, because oxygen is a measure of how much the cells are actually using energy for their cellular processes. And when we looked at our sort of SERCA efficiency measures, we used calcium uptake and SERCA activity measures. Our calcium uptake measures give us sort of a direct measure of how much calcium is being taken up into the cell, where the SERCA activity looks at ATP hydrolysis directly, and put them together, and we can actually look at our coupling ratio, which is how much calcium per ATP hydrolyzed. And lastly, we use some Western blotting in order to sort of quantify some of those proteins of interest, like our uncoupling proteins, sarcolipin and neuronatin, for example. Moving on to our adipocytes, similar methods for respiration were used. I'm just looking at cellular energy expenditure, and we performed some Western blotting to actually look at things like UCP1 content as well. So now getting into some of our results in our C2C12 cells first. When looking at figure A, what we can actually see is a significant increase in the inhibitory phosphorylation of GSK3 within the lithium treated group. And we see this in phosphorylated GSK3 alone, as well as the phosphorylated to total GSK3 ratio. We also actually noted some increases in PGC1 alpha within the lithium treated group. And this is a mitochondrial biogenesis marker. So we did sort of look a little bit further at mitochondrial markers, although we did not see any changes in any sort of other mitochondrial markers between the control and lithium groups. But when looking at our cellular respiration or our oxygen consumption, again, as our measure of energy expenditure, we do see that within the lithium treatment group, our energy expenditure or oxygen consumption was significantly increased. So in order to determine if this was due to SERCA, we could actually look at SERCA's percent contribution. So essentially when we treat with magnesium, this sort of stops SERCA in a way. So we can actually see how much SERCA is actually contributing to respiration. And sure enough, we actually do see that SERCA's percent contribution to basal respiration was increased within the lithium group. And here, this was actually met with significant reductions in calcium uptake in our lithium treated cells with no change in SERCA activity, which then led to reductions in SERCA's apparent coupling ratio. So this reduction in coupling ratio and SERCA's percent contribution sort of gives us an idea of maybe why we saw that increase in oxygen consumption. So when we did look at our proteins of interest, interestingly, we didn't actually see any increases in sarcolipin or neuronatin protein content in the lithium versus control groups, but we did see an increase in RYR1. And RYR1 is this calcium leak protein. So essentially, if you have more leak from the storage site, you're going to have more uptake by SERCA. So this futile cycling could also explain why we did see reductions in SERCA's coupling ratio. So overall, in our muscle cells, we do see that lithium treatment inhibits GSK3 and increases our cellular respiration, promoting SERCA uncoupling due to that lowered coupling ratio. So now looking at our cells for the adipocytes, again, similar to our muscle cells, we do see that this dose of lithium treatment does inhibit GSK3, as indicated by our increase in GSK3.
We lost sound from your end. We're having some technical issues.
Is it okay now?
Yes, we can hear you now.
Sorry about that.
No worries.
Okay, so yeah, so we did see that increase in GSK3 phosphorylation or inhibition of GSK3 in our lithium group. And then when examining, again, our oxygen consumption or energy expenditure, similar to our muscle cells, we did see increases in cellular respiration within the lithium treated cells. When looking at our proteins of interest, so UCP1, we do see increases in UCP1 content within our lithium treated group in the adipocytes. And like the muscle cells, we do see an increase in PGC1, that mitochondrial biogenesis marker. We also looked at mitochondrial footprints, although these were unchanged within our control and lithium groups between. So overall, similar to our muscle cells, we do see inhibition of GSK3 at this dose of lithium, along with increases in cellular respiration and increases in UCP1, which could have been indicative of why we saw that increase in cellular respiration. So now we can take a look at some of our in vivo data. So here we had two groups of our mice. So we had our control and lithium treated group, which was fed that 10 milligram per kilogram body weight per day dose. And this is where we actually were able to take some measures with the small animal DEXA. And we were actually able to look at body composition measures like lean mass, fat mass, and overall body weight. And we were also able to take our measures of energy expenditure using our metabolic caging system, which monitors activity, oxygen consumption, food and water intake. And together with our measures of lean mass and fat mass, we were able to actually normalize these energy expenditure measures to these different body compositional measures. So those were our sort of in vivo measures there. And then we did perform a number of ex vivo measures in both muscle and adipose. So our muscle measures were very similar to what we performed in our cellular model for muscle. And then our adipose measures were also similar with the Western blotting, but we did also look at histology of the adipose tissue in order to see if there were any sort of morphological or phenotype changes within those adipocytes after lithium treatment. So here are some of our in vivo measures from the study. So we actually did see that under 12 weeks of a normal chow diet, lithium supplementation did actually not alter body mass between groups, although lithium treatment did actually appear to increase food consumption. And we do see a significant difference in cumulative food intake by week 12 compared to controls within the lithium group. And furthermore, using our DEXA, our body composition analysis of fat mass showed no significant differences between lithium and controls. And similarly, we saw no notable changes in lean mass or muscle mass between the groups. But with that being said, there was an apparent increase in food consumption without an increase in body mass or any body fat percent changes. So this sort of suggested to us that there was actually an increase in energy expenditure with lithium supplementation. So we did actually observe that across light, dark, and overall daily periods, when normalizing our energy expenditure to both lean mass and fat mass, our lithium supplemented group did actually have increased VO2 or increased oxygen consumption, indicating increased energy expenditure. So importantly though, this increase in energy expenditure was not met with increases in cage ambulation. So they were expending more energy, but they weren't actually performing more activity. So this sort of suggested to us that there must be some sort of cellular mechanism that was actually increased in energy expenditure. So in order to investigate, this increase in energy expenditure that we did see in vivo, we performed our ex vivo measures on the soleus muscle. So here we see under this normal chow diet, our Western blot analysis showed that we have increases in the phosphorylation of GSK3 relative to total GSK3 within the lithium treated group. So we are seeing GSK3 inhibition. And when measuring our calcium uptake rates, we do see that when looking at them separately, calcium uptake and SERCA activity were unchanged between control and lithium groups. But when looking at them together, we actually do see significant reductions in SERCA's apparent coupling ratio within the lithium treated group. So the promotion of the SERCA uncoupling was actually met with a significant increase in the uncoupler neuronatin, but not sarcolipin. So overall, this sort of did shed light on that potential underlying cellular mechanism contributing to the increase in energy expenditure in vivo. And we can attribute that to inhibited GSK3 and SERCA uncoupling potentially through neuronatin. So now taking a look at some of our adipose data. So we did actually see in IWA or white adipose tissue, significant increases in phosphorylated GSK3. Although when we look at our brown adipose tissue, we actually don't see any inhibition of GSK3 here. Additionally, when looking at white adipose tissue, we saw significant increases in the lithium groups of UCP1s, that uncoupling protein, as well as some mitochondrial markers, including cytochrome C, pyruvate dehydrogenase, and PGC1 alpha. But again, when looking at our brown adipose tissue, it didn't appear that this thermogenic program was activated because we didn't see any changes with the lithium group compared to controls across any of our proteins of interest. So with that being said, we kind of looked at histology, and our histology actually revealed that GSK3 inhibition with the lithium supplementation actually leads to a significant beiging effect within the white adipose tissue. And it sort of took on this multiocular-like phenotype, so larger lipid droplets and more dispersed mitochondria. That's very typical of brown adipose tissue. So overall here, it appears that in adipose, the GSK3 inhibition actually turns on this sort of thermogenic program within the white adipose tissue, allowing for this almost beige phenotype to be seen. So what we wanted to look at was, is thermogenesis actually activated under high fat feeding? Because those measures were performed in chow-fed studies, but high fat feeding is actually a condition that would potentially benefit from thermogenesis or that increase in energy expenditure. So when examining our high fat measures, we did similar measures of body mass and food intake as we had in chow. Unfortunately, we didn't have our metabolic caging system or the DEXA machine at this time, but we still found some cool stuff. So here we can see that there was no differences in body mass between the high fat diet and high fat diet lithium group. Of course, we do see differences with that high fat feeding compared to control, but the fact that the lithium group was no different than high fat diet was kind of interesting. And sure enough, when we looked at our cumulative food intake, similar to our chow conditions, we actually saw that the high fat diet lithium treated group was actually eating more than control and high fat diet alone. So again, with this sort of increase in food intake, but no change in body mass, there must be some sort of cellular or energy expenditure mechanism at play here. So sure enough, we did examine our soleus muscle, and here we did actually see inhibitory phosphorylation of GSK3 within the high fat diet lithium group. And if you look at our representative, you can actually see it's almost like a sort of restorative effect to our low fat diet conditions when looking at the three groups there. And then we did also see increases in PGC1 alpha within our high fat diet lithium supplemented group. And when looking at our measures of SERCA inefficiency, we do see that in the lithium supplemented group, we have significant reductions in calcium uptake, no changes in SERCA activity, and therefore reductions in SERCA's apparent coupling ratio. So when we do see or take a look further at our uncoupling proteins, we actually see that sarcolipin and neuronatin are both significantly increased within our lithium fed group. So overall, this kind of shows us that lithium does inhibit GSK3 and promote SERCA uncoupling potentially through sarcolipin and neuronatin when placed on that high fat diet. In contrast, with our muscle and then our chow-fed adipose, lithium supplementation actually did not lead to any alterations in white adipose or brown adipose tissue within the high fat diet fed mice. So we saw no changes in GSK3 phosphorylation between groups. So what we thought here is potentially that lithium supplementation could not overcome the overactivation of GSK3 seen with this high fat feeding in the adipose tissue. So when measuring our mitochondrial protein content in both white adipose and brown adipose tissue, we didn't really see too much. We saw a little bit of a reduction in some of the mitochondrial proteins within white adipose tissue, but in brown adipose only changes in citrate synthase. And when we looked at our histological analysis, unlike the chow conditions, we didn't actually see sort of any beiging effect with lithium supplementation. So overall here, under the high-fat diet conditions in adipose, lithium could not quite inhibit GSK3 and therefore activate that thermogenic program in IWAT or BAT. So we are also aware that lithium could have sort of effects outside of GSK3 inhibition that could have potentially been activating thermogenesis. So to sort of establish whether there was a true connection between GSK3 and uncoupling proteins in our muscle, we actually generated a muscle-specific knockdown model of GSK3. And just a quick shout out to Brianna for doing that. She put a lot of work into sort of generating and keeping up this colony for our lab. And it's going to provide us with a lot of great results now and in the future. So when looking at our GSK3 knockdown model, essentially our GSK3 gene is less expressed. So without surprise, we actually do have a reduction in GSK3 content. So here we looked at our coupling ratio results. And again, separately, we didn't actually see any differences between the knockdown and wild type group. But then when looking at our coupling ratio together, we do see significant reductions in our GSK3 knockdown groups in coupling ratio. And then when we actually looked at our proteins of interest, we do see that in our GSK3 knockdown, we have increased sarcolipin and neuronatin content. So this is similar to our inhibitory findings with lithium. This lower coupling ratio is potentially due to increase in sarcolipin and neuronatin. And here, this sort of gives us some novel information establishing a real connection between GSK3 inhibition or knockdown in this case, and the expression of sarcolipin and neuronatin uncoupling proteins. So just to kind of sum everything all up, essentially what our sort of paper and our findings found was that low-dose lithium actually does inhibit GSK3 and increase the expression of uncoupling proteins, neuronatin, sarcolipin. And this is met with SERCA uncoupling in our skeletal muscle. And the GSK3 inhibition can actually promote this sort of beiging effect within the white adipose tissue. And overall, we do have sort of a new potential cellular target for offsetting the phenotype of obesity. So we do have some future directions using our small animal DEXA. For example, we know that the hormone estrogen has a number of metabolic implications. And right now, many of the studies that examine some of the measures of thermogenesis have been done solely in male mice. So as part of my PhD project, I will sort of be evaluating the effects of GSK3 inhibition on muscle and adipose thermogenesis in a female mouse model to sort of expand on this knowledge gap. And utilizing the Syntheta small animal DEXA for these studies is going to give us some really critical information on these in vivo measures of energy expenditure and body composition changes within our female model. So just wanted to say a quick thank you to obviously my supervisor, Dr. Fajardo, as well as the Fajardo research team and all the contributors to the JVC paper, because it was a long time coming and couldn't have done it without everyone's support. And Dr. McPherson as well played a huge part of that. And thank you to Syntheta. I'm happy to answer any questions.
Thank you for the presentation, Mia. We will now move on to the Q&A section, with the first question actually being, does SERCA play a role in adipose thermogenesis or just skeletal muscle?
Yeah, so that's a great question. So SERCA is actually expressed in the adipose tissue. It is a different isoform. It's SERCA2b rather than 2a, which is found in the muscle. But SERCA in the adipose tissue, as well as RYR, if you remember that calcium leak protein from the cellular model, actually can sort of have this futile cycling going on within the adipose tissue of calcium. So it does play a role for sure.
Thank you for the answer, Mia. The second question is, why do you normalize the energy expenditure data to body composition data?
Yes, so when we look at our energy expenditure, it's definitely important not just to take it as a whole, but to actually look at our body composition data as well. So, for example, if you look at lean mass or skeletal muscle is a very highly metabolic tissue. So whether it's through, certain calcium pumps like SERCA, or like the sodium pumps, or even just muscle contractions, muscle tissue actually plays a very high role in our total energy expenditure. So if you have increases in muscle mass, it's important to account for that increase because that could be playing a role into how much energy you're actually expending. So when you're normalizing to that sort of data, it gives you a bit more of an accurate measure of where that energy is coming from.
Thanks again, Mia. The next question is, how is lithium transported into adipocytes, myocytes?
How is lithium transported?
Into adipocytes and myocytes. You can also see the questions asked in the Q&A box.
Oh, I see that.
Sometimes I don't pronounce it too well, so yeah.
No, that's okay. That's actually a good question. I'm not sure if there's like an actual transport measure, or like a transport channel or something. I should probably know that, but I think it is like a permeable um, molecule, similar to like a salt, because it is lithium chloride that we're giving them. So it'd be similar to like a potassium chloride or a sodium chloride.
Thank you, Mia. So we'll move on to the next question, which is, why is it important to normalize energy expenditure to body composition measures?
I think I answered that one
Oh,
from
yeah.
the previous question, but yeah, just again accounting for that that change in muscle mass because it is a highly metabolic tissue. So
Okay, so the next one is, why did you choose the dose you did for cells and for the mouse model?
yeah, so starting with our cells, we did have some previous work from um, another PhD student in another lab that collaborated with us at Brock. And he showed that inhibition of myoblast fusion and GSK3 and myoblast fusion was shown with a 0.5 millimolar dose. They did try to give that lower dose that we use in vivo to those myoblasts or the cell line, but it didn't actually result in inhibition of GSK3. So the cells did require a little bit of a higher dose. And for the 10 milligram per kilogram per day that we used for the in vivo, we actually sort of piloted that from a different paper looking at atherosclerosis. And it showed that there was inhibition at that dose with like their model that they were using. And what they showed was that it actually sort of attenuated the obese phenotype and weight gain at that dose as well. So we sort of piloted that with GSK3 and we did see inhibition at that dose.
Thank you, Mia. I'll move on to the next one, which is, how can lithium be used as a treatment in human application?
Yeah, so lithium has been used in the treatment of bipolar disorder, although it is used at higher doses for those, for the, what is clinically used for right now. But what we're sort of trying to push is a lower dose level to see some of those more beneficial effects. And we do actually have a study that shows trace levels of lithium, so very low dose amounts in water, actually negatively correlates with the sort of population obesity. So there's less obesity with higher trace amounts in certain areas. So yeah.
Thanks again. Are there other ways to look at GSK3 activity besides phosphorylation status?
Yes. So we could actually also perform a GSK3 activity assay, which our lab has done for other models. So it'd be kind of interesting to see if that phosphorylation, um, is correlated with the inhibition or inactivity. But we can actually perform a GSK3 activity assay as well.
Perfect. And the next question is, why target cellular methods to increase energy expenditure, and why not just exercise?
That is a good question. Unfortunately, exercise is, sort of our best way to increase our overall energy expenditure. It doesn't take very much, but it actually has been shown, Stats Canada showed that only 2 out of every 10, I believe, Canadians actually meet the recommended physical activity guidelines, which is not very much. I think it's only like 150 minutes per week. So it kind of shows the need to have some alternative ways to promote this energy expenditure. And exercise sometimes is not always feasible. There's societal and socioeconomic barriers to access to exercise as well. So there's that sort of factor in there.
Okay, perfect. Thank you. So we've reached the end of our session today, and to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we will be sure to answer any questions in the written transcript, and we'll work to get this out to you over the next few days. I would like to thank Mia for the great presentation today, and I trust that we have provided you with a better understanding of how low-dose lithium supplementation promotes adipose tissue browning and uncoupling in muscle and mice. If in the days and weeks to come you have further questions about the modalities discussed today, I encourage you to reach out to us here at Syntheca, and we'll be happy to discuss them further. Thanks again to all of you for taking time out of your day to attend our session, and we look forward to seeing you at a future Syntheca event. Have a great rest of your day.